Hi, everyone, and thanks for joining us. I'm Dr. Stephanie Call, your moderator for this event. It's really exciting to see people logging in from all over the globe. So whether it's the beginning of your day or you're winding down for the evening, I hope you find this educational session informative and at least a little fun. Today, we're going to have a conversation about dynamic light scattering, or DLS, and how it can be a really useful tool for learning more about your protein samples. Anyone from a basic science researcher engineering new proteins to a downstream pharmaceutical researcher doing QC on a final product will hopefully see the benefits of DLS. A few housekeeping notes before we get started. You'll see a panel at the bottom of your screen with a few icons. There is a slide explaining these icons and how to use them that will be visible throughout the talk unless you minimize it. The media player is the icon in red. That's where you'll be viewing this session. If at any time during the talk you think of a question, select the purple Q&A box and enter a question. You'll see it disappear once it's submitted, but don't worry, we can see it on our end. We will address all of these questions at the end of the talk. It looks like we have a lot of attendees, so if we don't get to your question during the recording, a specialist will be in touch with you later. With that, I would like to introduce Dr. Leah Pandisha. She's a product specialist at Nanotemper, and she's going to cover the theory, the measurement setup, the data interpretation, and some examples of how DLS can be used in order to enrich your protein research. Thank you again for joining us, and I'll turn the floor over to Leah.
Great, thank you, Stephanie. All right, so let's get started. So proteins form three-dimensional structures, each with their own characteristic properties such as size, molecular weight, preferential folds, etc. And as proteins interact with other molecules in their environment, these properties can change, which directly affects their functionality and stability. And in particular, the changes in their structure or folding can lead to changes in their size and can induce protein polymerization. As with most things in life, there are pros and cons to these changes. So either the biomolecular machinery carries out its intended function and our cells, and essentially we as humans, continue to exist, or the misfolding of these molecules induces disease-related aggregates, which we must combat with therapeutic strategies. Because protein aggregation is mostly irreversible, being able to characterize the pathway to the aggregated state to avoid this potentially deadly and costly phenomena is critically important. And of course, further elucidating the less common pathways these proteins take also allows researchers to better understand their complex functionality and use this to their advantage. While there are numerous biophysical techniques used to probe protein stability, today we'll be discussing a light scattering technique, dynamic light scattering, or DLS, to provide detailed information about the size and quality of particles in solution. So starting with the basics, what is light scattering? Light scattering occurs when electromagnetic radiation or light impinges on a molecule and leads to a polarization of its charge. Unlike absorption, which requires a change in the energy states of the molecule, in light scattering, the incident light is deflected off the particle without affecting the molecule's energy. There are two main types of scattering relevant to DLS, Rayleigh and Mie. Both types of scattering are elastic, meaning that the energy of the incident light doesn't change when scattered. So if the incident wavelength is 600 nanometers, the light scattered is also going to occur at 600 nanometers. This also means that since energy is inversely proportional to the wavelength, the lower the incident wavelength, the more energy, and therefore the more scattering intensity you get. Rayleigh scattering occurs when the particle impinged upon is smaller than 1/10 of the incident light's wavelength. The scattered light is isotropic, or it scatters equally in all different directions, and there's no change in the intensity depending on the angle of observation. Mie scattering, on the other hand, occurs when the particle size is larger than 1/10 of the incident wavelength. Mie scattering is angle dependent, so the back reflected light is less intense than the forward scattered light. And the larger the particle size, the stronger the angular dependence is, and the more intense that forward scattered light is. Light scattering is a non-invasive technique that can be measured in solution, making it an ideal bioanalytical tool. Different light scattering techniques provide different information about a particle, like its size, mass, diffusion coefficient, concentration, and charge, but all are extremely sensitive to the presence of small amounts of aggregates, making it a highly sensitive technique. While there are different light scattering techniques, today I'm going to be focused on dynamic light scattering, or DLS. So what is dynamic light scattering and how does it work? DLS measures the time-dependent fluctuations of scattered light intensity due to the Brownian motion of particles. Brownian motion is the random movement of particles in solution resulting from their collisions with the surrounding molecules. The larger a particle is, the slower it's going to move or diffuse through solution, while smaller particles are going to move faster. The speed that these particles are undergoing Brownian motion is dependent on the particle size, the sample viscosity, and the temperature. And the rate of these measured intensity fluctuations is dictated by a particle's movement in solution and described by the diffusion coefficient d. This equation here is the Stokes-Einstein relationship, where the diffusion coefficient is inversely proportional to a molecule's hydrodynamic radius r, and can therefore be used to calculate a particle's size. So the hydrodynamic radius is defined as a hypothetical hard sphere that diffuses at the same rate as the particle being measured, which means it's an apparent radius, not an absolute measurement. Of course, the particle's core size is going to affect the hydrodynamic radius. However, any factors that influence its diffusion through solution will also affect this observed radius. And the more drag that a particle has as it moves, the slower it's going to diffuse and the larger that apparent radius. So for instance, a particle's shape is going to influence this diffusion. So as a protein structure changes as it unfolds, the hydrodynamic radius will increase with increasing surface area of that molecule. However, for any non-spherical particles, DLS outputs the radius of a sphere having the same average diffusion coefficient as the measured particle. Changes to the surface of the particle are also going to affect the apparent radius. So if there's any additional polymer linkers, that will reduce the diffusion speed of the particle and increase the radius. The addition of ions and changes to the ionic strength can also affect particle diffusion by altering the thickness of the electric double layer. So the larger the ion concentration, the more compressed the double layer of ions around the particle will be, which increases the speed of diffusion and decreases the hydrodynamic radius, while lower ion concentrations are going to expand that electric double layer, decreasing the diffusion of the particle and increasing the radius. And then lastly, concentration-dependent particle interactions can force particles to clump together, which also increases their apparent radius. So DLS can probe a wide range of particle sizes, typically down to 0.3 nanometers up to 10 micron in size. But proteins and most biomolecules tend to be considered small on this scale. The average radius of IgG is 7 nanometers, lysozyme is 2, while larger particles like virus-like particles are 55, smallpox virus is 350, and then on the higher end you have a cancer cell, which is 1 micron. And even as proteins unfold and form oligomers, they still fall on the smaller end of that particle sizing spectrum. As with all instrumental techniques, there are some physical limitations to DLS. So I mentioned earlier that Rayleigh scatter occurs when the particle size is less than 1/10 of the incident light wavelength. And this scattering intensity is going to be proportional to the particle diameter to the 6th power. So that even these small particles like lysozyme is going to scatter a significant amount of light. However, that also means that a 50 nanometer particle is going to scatter 1,000,000 times more light than a 5 nanometer particle, making it difficult to measure small particles in the presence of a high concentration of larger particles. But this is precisely why DLS is so sensitive to the presence of aggregates in solution. And to look on it in a positive light, DLS will detect even small aggregate concentrations present in large populations of smaller protein particles. DLS is also a relatively low resolution technique. So for example, while you cannot differentiate between ratios of monomers to dimers in a sample mixture, you can, however, detect that the sample is a mixture composed of different species. So how does DLS work? A DLS instrument consists of a coherent light source or a laser that's directed into the sample volume and backscattered by particles in solution. This backscattered light is collected by a detector that is set at a specific angle to the sample volume. And the closer this detector is to the incident angle, the larger that Rayleigh scattering contribution will be, and the less intense the Mie scattering contribution will be, since Mie scatter is more intense in the forward direction. So the detector angle coupled with our laser wavelength increases the instrument's sensitivity towards smaller particles like proteins and other biomolecules. DLS is an ensemble technique, meaning that the output from the acquired measurements is a superposition or an average of many particles. So the incident light is going to be focused on a small volume of sample through which the particles are going to diffuse. And each particle in this ensemble scatters light, and the sum of each particle's electric field vectors describes the strength and directionality of the electromagnetic wave. As these particles in the scattering volume move and bump into each other and other solvent molecules, they inevitably are going to end up in a different location from their initial position. And this movement is going to result in changes in the direction in the scattered light and the direction that it's traveling, as well as phase differences between the light waves. And the changes in the phase interference patterns, like the constructive or destructive interference, are reflected as fluctuations in the intensity of the scattered light over a time that the particle is diffusing through the solution. So here we are looking at the intensity fluctuations of scattered light as a function of time, where IT is the intensity at the initial particle location, and IT plus tau is going to be the intensity at a location after a period of time tau. The smaller a particle is, the faster it's going to move and diffuse compared to a larger particle. So smaller particles are going to have more collisions, and they're going to change locations more frequently than larger particles. And these collisions and movement translates to more scattered light intensity fluctuations. The autocorrelation function is going to extract these time-dependent intensity fluctuations and compare the similarity of the signal intensities. So this equation shown here describes this autocorrelation function g squared, and you can see the dependency on the intensity over time and the parameter q, which is the scattering vector and takes into account instrument parameters and sample properties. So at the initial position, the signals are pretty similar, regardless of the particle size. The autocorrelation function or autocorrelation value is going to be quite large. So it's in our instrument roughly 1, which we see at the initial time point IT or time 0 in both of these plots here. The further that time goes on, the larger the difference between the intensity fluctuations, which decreases the similarity between the intensities and therefore that autocorrelation value. So this is going to decrease as a function of time for both large and small particles. However, the larger particles are slower, so that difference between the intensity at the initial location 0 compared to the intensity at the location over time is going to be relatively small, and those intensities are a lot more similar. The autocorrelation values as functions of time are plotted to get a decay, where these small particles have an autocorrelation function or an ACF that decays rapidly, while the larger particles have a slower decay. And the rate of this decay is described by the diffusion coefficient. So how can we interpret the autocorrelation function or the ACF to obtain particle sizing information? The autocorrelation function is the raw signal that is fit with an algorithm to determine the diffusion coefficient, which we need along with the temperature and viscosity to calculate the hydrodynamic radius via the Stokes-Einstein equation. The autocorrelation function not only elucidates the mean size of a particle determined from the time that the decay starts, but also its homogeneity indicated by the gradient of the decay. And the ACF is also going to provide information and clues to the sample quality, so whether or not the data is good enough to do a reliable analysis. And there are 2 ways of analyzing the autocorrelation function to determine the particle size: the cumulative analysis and the size distribution analysis. So the cumulative analysis is defined by ISO standards, and it fits a single exponential decay to the ACF and assumes a Gaussian intensity distribution. It assumes a single particle species that has a single rate of diffusion so that the output is a single average diffusion coefficient, and therefore we get an average hydrodynamic radius. The cumulative fit to the raw data is shown on the top right. So the raw data is in gray and the cumulative fit is overlaid by the green curve. The average hydrodynamic radius is indicated in the size distribution plot on the bottom right by this green line here. And the cumulative fit also looks at the width of the distribution for the entire sample volume to provide information on the homogeneity of the particles described by the polydispersity index, or PDI. So the second fit is the size distribution fit, and this uses an intensity-weighted distribution of multiple diffusion coefficients to account for different populations of sizes and can therefore calculate more than one particle size. This algorithm is going to identify different populations in the scattering signal and then plots the relative frequency of the various size groups. And since it doesn't assume a certain type of distribution of the diffusion properties, it's more suitable for polydispersed systems. So we can see in the top the raw data, which is fit by the size distribution fit in blue, and in the size distribution plots on the bottom right, the radii are displayed as the blue peaks that correspond to different population sizes. As I alluded to earlier, the polydispersity index is a measure of the heterogeneity of the particle sizes and can be used as a metric for sample quality, indicating whether there are multiple species present in the sample volume. So the more polydisperse or the more particle species present, the higher that PDI value will be. And this is further seen in the size distribution plots, where the wider the peak, the more polydispersed the sample is. So the bottom plot, we have a relatively wide peak compared to the top plot, and it has a PDI of 0.29, indicating a polydispersed sample, where the top plot has a sharp, narrow peak with a PDI of 0.17, indicating a monodispersed sample. So samples with a PDI less than 0.25 are considered monodispersed, whereas particles with a PDI greater than 0.25 are considered polydispersed, and the greater that value is, the greater the likelihood of significant aggregate formation. So since protein engineering or formulation changes can potentially alter a protein's behavior and stability and lead to aggregation, having a better picture of how these changes affect the protein's size and polydispersity allows researchers to make better decisions about what molecules to move forward with or which molecules to avoid, saving both time and money. So here we have some DLS data of NIST-MAB in histidine buffers shown by the navy plot, and bovine gamma globulin in saline buffer shown by the purple plot. So the size distribution plot indicates that MAB is very homogeneous and monodispersed, indicated by a small PDI value and a sharp, narrow peak. BGG, which is a fraction of blood serum that consists of whole antibodies and non-albumin plasma proteins, in this example shows that it's not in its optimal buffer. It's much less monodispersed than the MAB sample, indicated by the larger PDI value of 0.2. And if we look at the size distribution plot, we see a broad tailing peak reflecting a wider size distribution. So obviously selecting the best storage formulation and storage buffer is necessary to make sure your sample is stable. And we want to optimize those storage and assay buffers to make sure that we have stability and functionality for a long period of time. So here we are looking at an accelerated stability study with Herceptin in two different formulation buffers, histidine and sodium acetate, to identify which formulation retains the protein's monomeric state. The samples were measured before and after incubation at 80 degrees for 14 hours. Proteins that are tightly folded and stable are going to give a single sharp peak, which is consistent with the size distribution plots on the top for both formulations prior to incubation. While Herceptin remains stable in sodium acetate buffer after incubation at high temperatures, indicated by this purple peak here that doesn't differ very much from the size distribution peak prior to incubation. On the other hand, in the histidine buffer shown by that navy peak, not only does the radius increase in size when we see a shift to larger radiuses or radii, indicating sample oligomerization, but the peak also broadens, reflecting an increase in the PDI value in the sample polydispersity. DLS measurements can also be used to confirm binders from primary high-throughput screening assays. So drug discovery groups often have huge libraries of compounds to screen, and no method is 100% reliable in avoiding either false positive or negatives. However, binding of small molecules often induces structural changes to the target protein, which alters its size. And by monitoring the hydrodynamic radius of the protein before and after complexation, subtle changes in that RH value can be screened to determine binders. So here we see the crystal structures of heat shock protein 90 alone and in complexation or bound to ATP. So not only does the binding increase the thermal stability of the protein shown by the nano-DSF data on the left, but binding also induces a change in the conformation to a more compact state indicated by the crystal structure data, which is also reflected in the RH plots on the bottom right. So we know that the size of a particle affects the amount of scattered light intensity and therefore resolution, but the concentration of particles is also going to affect a DLS measurement. So in particular, high particle concentrations can result in interparticle interactions affecting the apparent radius. And the concentration-dependent interparticle interactions can be evaluated using the self-interaction parameter KD. This is also referred to as the diffusion coefficient parameter and not to be confused with the binding affinity. So since the diffusion coefficient of a protein is an intrinsic property dependent on the temperature of the solution and the size and shape of the protein, if the diffusion coefficient changes as the concentration of the protein changes, this is going to indicate that there are interactions between the protein, which is affecting its diffusion. So by plotting the diffusion coefficient as a function of concentration, we can then determine whether the protein favors self-association, where the attractive forces between protein molecules slows down the diffusion coefficient, giving a negative KD value, or repulsive interactions, increasing the diffusion coefficient and resulting in a positive KD value. So the KD can be used to understand self-interaction at dilute concentrations and then extrapolated to predict behaviors at higher concentrations. So for biologics groups, predicting the propensity for self-interaction is critical to selecting ideal candidate molecules for successful formulations. And here we see the effect of two different formulation buffers on the self-interaction parameter. So histidine buffer shown by the navy data increases in the diffusion coefficient with increasing concentration, indicating repulsive forces between the protein molecules. Once salt is added to the formulation buffer, that diffusion coefficient decreases with increasing concentration, indicating attractive forces, where the addition of salt ions could potentially be shielding charges on the surface of the protein molecule, reducing the electrostatic repulsion between molecules. Regardless, the addition of salt to the formulation buffer promotes self-association of the protein, which could lead to aggregation, producing a less than ideal candidate molecule for a therapeutic. So to summarize DLS in a nutshell, researchers ultimately want to know the particle size and understand the size of the particle. And dynamic light scattering can be used to ascertain the rate of the diffusion of molecules in solution by looking at changes to the interference pattern of light being scattered by these particles. So since particle size determines how fast it moves and diffuses through solution, that affects the rate of these intensity fluctuations of the scattered light. And by looking at the rate of the fluctuations, we can then correlate how quickly the fluctuations are occurring to derive the autocorrelation function. And the autocorrelation function is fit to extract a diffusion coefficient from the rate of decay. And using the Stokes-Einstein relationship, knowing the viscosity and the temperature of the solution, we can then calculate the hydrodynamic radius of a particle. So if at any point during this talk you thought to yourself, well, I know my molecule has a relatively high melting temperature, but we haven't looked at the colloidal stability, or it would be really helpful to check for oligomerization of my sample prior to my cryo-EM experiments, then this sneak peek into the Prometheus Panta is going to be right up your alley. So with Prometheus Panta, thermodynamics can be characterized by monitoring protein unfolding. Large aggregation formation can be better understood through turbidity measurements, and now particle sizing can be assessed with the addition of the DLS optics. And all this information is easily obtained and analyzed using our control and stability analysis software. So please join us for our Panta launch event on March 30th. The link to register for this event, as well as an e-demo event of the Panta on March 17th, are listed in the resource section of the toolbar at the bottom of your screen. Thank you for your attention, and I would be happy to take any questions you've been sitting on for the past 20 minutes.
Okay, well, Leah, thank you so much for that talk. It was really informative, I think. And with that, as you can all see, it's time for our Q&A session. So I have several questions collected by everyone who's attended today. So Leah, let's get started. The first question that we got from a couple people, does the scattering angle affect the data collected by DLS?
Yeah, that's a great question. So there are three things that are going to affect that scattering angle and the intensity of the scattered light. So the scattered angle, the wavelength, and then the size of the particle. So the scattering angle generally has the least impact on the intensity. More important is going to be the size of the particle, which, as I mentioned, is going to scale the intensity by a factor of 6. The wavelength of light used to scatter light off the particle can also have an effect on the intensity, where shorter wavelengths are going to be more intense. However, having the detector at a backscattering angle orientation also is going to increase the sensitivity to smaller particles. So there are a couple things that are going to affect that absolute scattering intensity.
Always plenty of variables in every scientific experiment. Okay, another question, which is a little bit more of a practical question. How much sample do you need to run a DLS experiment?
So many instruments are going to work with a few microliters of sample. There are required minimum concentrations, roughly between 0.2 and 0.5 mg per mil. However, that's going to depend on the size of the particle that is actually scattering that light. So the larger your particle is, the more scattering intensity you get, so you could use lower concentrations of sample.
Right, okay. The next question's kind of like two parts. We have sort of a couple combination questions from a few people. So does this work on nucleic acids, or is it only for proteins? And then kind of along that line, what is the smallest protein that you can measure?
So yes, you can most certainly use DLS for nucleic acids and proteins. Basically any particle that's going to scatter light you can use DLS for, which is pretty awesome. So like, for instance, if you have sugars in your buffer, you'll actually see that sugar peak in the DLS data as well. And then in regards to the size of the particle, with DLS, the lower limit is typically 0.5 nanometer hydrodynamic radius.
Cool. Great. So another kind of experimental question. Are there any limitations with anything that you can add to the protein, like buffer additives or anything else in the formulation?
So generally, no. That's one of the great benefits of DLS is that it can actually show you how your buffer formulation affects the oligomerization of your protein. For most DLS experiments, you're going to be working at room temperature initially. So it is useful to optimize that storage buffer of your protein so it's stable at room temperature for a specific amount of time. Large particle additives like sugars or PEG are going to show up in the data. And then kind of just monitoring the sample at room temperature in those buffers is just useful for looking at the formulations.
Cool. All right. Here's kind of another question. This is similar to what was asked before, but is there anything else that people need to keep in mind when they're preparing a DLS sample, like how to prep the actual sample before putting it into whatever you're measuring it with?
Yeah, that's an excellent question. And it is important. And one of the really awesome things about DLS is that it can look at different particle sizes and really small particles. So because DLS is super sensitive to aggregates, it also means that it's going to pick up any little dust or dirt particle that you potentially have in your sample. So it's super important that your plasticware is clean, and that any pipette tips or vials or well plates or anything that you're going to prep samples in are stored and covered so that no dust or dirt from the environment potentially gets into there. It's also really good practice to centrifuge or filter your sample prior to measurements to remove aggregates. However, if you're doing an experiment where you are looking to monitor aggregation, then you wouldn't want to worry about that as much.
Right. So along that line, if you're looking at the data, how do you tell the difference between oligomerization and aggregation?
So oligomerization is going to lead to a hydrodynamic radius that is generally still in the same size range as single proteins. The autocorrelation function from the oligomerization is still going to translate into some of those distinct population peaks in the intensity distribution plots that I showed earlier. Aggregation data, on the other hand, is going to fit poorly by that autocorrelation function, and it tends to give a very high PDI value. So I also mentioned that, the closer the PDI gets to 1, the more aggregation formation you have, or the potential for aggregation formation. As you have large aggregates, you're typically going to have data quality that decreases. And then once your particles are reaching a certain size of aggregate, it's more appropriate to use other methods, like monitoring the turbidity of your sample to look at aggregation formation.
Right. Okay. It looks like we have a question about one of the slides you shared with the applications. So do all small molecule binders show a change in the hydrodynamic radius?
So that's a good question. Whether you see a change in the hydrodynamic radius is going to be dependent on the mechanism of binding. So how that binding significantly changes that hydrodynamic radius, and it's going to be relative to that unbound form. So some proteins, like in the heat shock protein 90 sample example I showed you, it bunches. And then when you have a binder, you get that more compact state. Some proteins, when you have a binder, they're actually going to expand in size and grow larger. So the best way, it's relative to your sample, the best way to determine if the conformational changes that you're seeing are actually significant and can be probed with DLS is just to do a positive control binder with the protein of interest.
Cool, thank you. We have one more question, it looks like. This is a theoretical question. Can you determine molecular weight from DLS?
So not really. There are some protocols in the literature where you can use standard curves to estimate the molecular weight based on some parameters, but it's a relatively loose assumption if there's a fixed density throughout the particle. So because that apparent radius of the particle is dependent on its shape, it's impossible to accurately determine a molecular weight of some unknown sample with DLS. However, if you used a calibration curve, you can kind of get a rough estimate of that specific oligomeric state of the sample. But there are also just other complementary techniques that you could use to look at the molecular weight in particular, if that is really your parameter of interest.
Right. No parameter, no technology gives you everything. So.
Exactly.
Well, thank you so much, Leah. This has been really informative, and I appreciate you helping me answer all of our viewers' questions.
Of course.
Thank you, the viewers, all of you, for sticking with us to this talk. I hope everybody has something of interest that they took home from this. Please check out the Resource Center. There's more links to future events we're hosting that might be of interest to you. And of course, if we didn't get to your question, we'll be in touch in the future. Thank you all so much for attending, and have a great rest of your day. Thanks.
Thank you.